You think you know DNA, double helix, blueprint of life, instructions passed from your parents written in 4 simple letters. That's what they tell you in school. But DNA is stranger than anyone imagined. 8% of your genome comes from viruses that infected your ancestors millions of years ago. Pieces of ancient plagues now running your cells. DNA folds into shapes that look nothing like Watson and Crick's ladder. Left-handed spirals. 4-stranded knots. Structures that resist destruction and control when your genes turn on. And scientists just found hidden switches buried in your chromosomes that let genes talk to each other across distances of nearly 1 million base pairs. The molecule storing your genetic code also behaves like both a solid and a liquid at the same time. It bends, it flows, it remembers. What we thought was junk turns out to be powerful machinery. What we dismissed as evolutionary debris is now rewriting the story of what makes you human. If you value documented genetics and molecular biology history like this, subscribe. It's the best way to support our work. Also, comment from which city you're hearing this from. This script is researched and factual. Using sources from the journal Nature, published July 2nd, 2025, detailing range extender elements discovered by Assistant Professor Evgeny Kvan and his team at UC Irvine's Charlie Dunlop School of Biological Sciences in collaboration with Lawrence Berkeley National Laboratory. And from the journal Science Advances, published July 2025, from Kyoto University's Institute for the Advanced Study of Human Biology, documenting ancient viral DNA's regulatory functions in early human development. Real peer-reviewed molecular genetics from labs actively sequencing and engineering mammalian genomes. Now back to the information. 8%, that number sits in your chromosomes right now, not genes for eyes or blood type. Viral DNA. Human endogenous retroviruses, HERVs, 60,000 separate pieces scattered through your genome. Relics of pandemics that swept through primate populations between 5 million and 100 million years ago. Retroviruses work by hijacking cells. They insert their genetic instructions directly into the host's DNA. Usually that DNA dies with the infected cell, but sometimes a virus hits an egg or sperm, a germ cell. And that viral code gets passed down, generation after generation. It becomes permanent, a genomic fossil. Scientists found HERVs in chimps, in gorillas, in macaques, the same viral sequences sitting in all of us. Proof these plagues infected our shared ancestors before we split into different species. Before there were humans, the viruses were already there, waiting, writing themselves into what would become you. For decades, geneticists called this junk DNA, useless baggage, evolutionary trash. Then, in July 2025, a team at Kyoto University published something that shattered that view. They focused on a family of viral elements called MER11, ancient sequences that came from retroviruses and copied themselves throughout primate genomes around 40 million years ago. The team developed a new classification system. Instead of lumping all MER11 sequences together, they traced evolutionary relationships. They divided the family into 4 subfamilies: MER11, G1 through G4, oldest to youngest. Then they tested nearly 7,000 of these sequences using a technique called lentiviral massively parallel reporter assay. They inserted each sequence into human stem cells, early neural cells, and measured how strongly each one activated nearby genes. The youngest subfamily, MER11G4A, showed massive gene activation potential. These aren't broken fragments of dead viruses. They're switches, enhancers. Regulatory elements controlling when and where specific genes turn on during the earliest stages of human development. The Kyoto team found something else. MER11 G4 sequences contain specific DNA motifs, short stretches that serve as docking sites for transcription factors, the proteins that control gene expression. These motifs differ between humans and chimpanzees and macaques. Each species evolved slightly different versions. That means these viral sequences didn't just sit passively in our genomes. They adapted. They changed. They became integrated into the regulatory networks that make humans different from other primates. A study published in Nature in October 2025 went further. Researchers used stem cell models called blastoids to study human embryos in the first few days after fertilization. They discovered that an endogenous retrovirus called LTR5H is absolutely essential for pre-implantation development. When they disabled this viral element, the blastoids failed to form properly. Cells couldn't organize into the structures needed to become an embryo. The virus wasn't junk, it was necessary. Specifically, LTR5H acts as a powerful enhancer. It boosts activity of nearby genes. One of the most important is ZNF729, a gene unique to humans. The viral DNA insertion acts as a master key activating this gene during the critical window when embryonic cells are deciding their fate. Without the virus, that gene stays silent. Without that gene, development stalls. This isn't a one-time fluke. The most famous example is syncytin, a gene humans and all other mammals use to build the placenta. Syncytin came from a retrovirus. The protein it makes originally helped viruses fuse with host cells. Evolution repurposed it. Now it helps the placenta fuse with the uterine wall. Pregnancy in every mammal depends on a protein that started as a viral weapon. Remove syncytin and pregnancy becomes impossible. The virus didn't infect us. It became us. Researchers at the University of Michigan found a Hervey family called K111 that was completely hidden in the human genome. David Markovitz and his team were studying blood samples from people with HIV when they found viral RNA that didn't match any known sequence. They searched the annotated human genome. Nothing. They checked the chimpanzee genome and found one copy. Then they went back and searched human DNA more carefully. K111 exists in hundreds of copies in modern humans, around 5 copies in Neanderthals and Denisovans, 1 in chimps. 0 in older primates. This virus entered the genome after humans split from other great apes, but continued replicating and spreading through our chromosomes. Most of it sits in the centromeres, the repetitive regions at the center of chromosomes that control how DNA separates during cell division. K111 provides a molecular barcode, a way to track and identify specific centromeres, which is crucial because centromeres are the last major unmapped regions of the human genome. The virus left a trail that scientists can now follow to understand chromosome structure. But viruses aren't the only strange thing hiding in DNA. The shape itself is wrong, not just the double helix. Other forms, non-BDNA structures that were supposed to be lab artifacts, G quadruplexes, 4-stranded DNA held together by guanine bases stacking into flat squares, zDNA. A left-handed spiral that twists the opposite direction from normal DNA, cruciforms, cross-shaped junctions formed by palindromic sequences folding back on themselves, I motifs, 4-stranded structures made from cytosine-rich regions. Scientists found all of these in living cells, in genomes, at specific functional locations. A study published in Nucleic Acids Research in April 2025 mapped these non-B structures across the complete telomere-to-telomere genomes of humans and 5 other great ape species. Katerina Makova at Penn State led the team. They used newly finished genome assemblies, complete sequences with no gaps. For the first time, they could see into the repetitive regions that were invisible in older drafts, centromeres, telomeres, the ends and centers of chromosomes. These regions are packed with non-BDNA motifs. The team found that different sequence patterns produce different structures. Alternating purine and pyrimidine bases favor zDNA. Stretches of guanines form G quadruplexes. The structures aren't random. They cluster at regulatory regions, at replication origins, at places where DNA needs to be opened or bent or marked for the cell to function. G quadruplexes showed up thousands of times in human chromosomes. They're enriched in gene promoters, the regions just upstream of genes where transcription starts. They also concentrate in telomeres, the protective caps at chromosome ends. Recent work shows G quadruplexes help control how telomeres are maintained, how chromosomes stay stable. A study in Nature, published in May 2025, found dental biofilms contain both G quadruplexes and zDNA in the extracellular matrix surrounding bacterial clusters. These structures resist degradation by DNase-1, the enzyme that normally chews up DNA. The non-canonical shapes provide physical protection. They make the DNA harder to destroy. Bacteria use them as shields, and the same principles apply in human cells. Non-BDNA is tougher, more stable in certain contexts. That stability has consequences. Some G quadruplexes slow down DNA replication. If a replication fork hits a G quadruplex, it stalls. That pause can cause mutations, breaks, genome instability. Multiple studies link G quadruplex motifs to cancer, to sites where chromosomes frequently break and rearrange in tumor cells, but G quadruplexes also regulate gene expression. They can block transcription or enhance it depending on location and cellular conditions. Evolution kept them because they're useful. Even though they're dangerous, zDNA is stranger still. It's a left-handed helix. Watson and Crick proposed a right-handed double helix, BDNA, the standard form, but under certain conditions, stretches of alternating purines and pyrimidines flip into zDNA. The backbone zigzags, the groove pattern reverses, and cells have proteins that specifically bind zDNA, ADAR1, an enzyme that edits RNA. It has a zDNA binding domain. When ADAR1 finds zDNA, it marks that region for regulatory attention, editing, silencing, activation. A paper published in September 2025 describes how non-canonical structures like zDNA and G quadruplexes appear in viral genomes, not just human chromosomes. SARS-CoV-2, hepatitis B, HIV, cucumber mosaic virus. These pathogens use non-BDNA to regulate their replication, to control when they activate, to hide from the immune system. Researchers found that drugs targeting viral G quadruplexes can inhibit replication. Small molecules that stabilize or destabilize these structures change how the virus behaves, which means the same strange DNA shapes humans use for regulation also serve as viral control switches. The viruses learned from us, or we learned from them. Now here's where it gets mechanical. DNA isn't just information, it's a physical object, a polymer, and polymers have material properties: stiffness, elasticity, viscosity. In August 2024, a team at Princeton led by Cliff Brangwyn published a method to bend DNA strands using light. They used laser beams to create liquid droplets called condensates inside living cells. These droplets attached to specific DNA sequences and pulled them around using surface tension, like tiny fingers plucking strings. What they found surprised everyone. Chromosomes behave like both an elastic solid and a liquid. Bend a piece of chromosome and it snaps back like rubber, but hold it there and it slowly flows and relaxes like honey. This dual nature is called viscoelasticity, and it matters because genes need to move, enhancers need to contact promoters. Chromosomes need to reorganize during development. If DNA were purely solid, it would be too rigid. If it were purely liquid, it would be too chaotic. The viscoelastic middle ground lets cells control organization precisely. The Princeton team measured how much force it takes to move parts of a chromosome. The answer depends on time scale. Fast pulls require high force. The chromosome resists. Slow sustained pressure requires much less. The chromosome yields, flows around the obstacle, reforms in a new configuration. If you stretch out all the DNA in one human cell, it measures about 6.5 feet long, 23 pairs of chromosomes, 46 total. Each one is a continuous molecule coiled and folded and wrapped around millions of proteins. The whole package fits inside a nucleus roughly 5 microns across. That's compression by a factor of hundreds of thousands. To achieve that level of packing, DNA wraps around proteins called histones. 8 histones form a core. DNA wins around the core 1 3/4 turns. That unit is called a nucleosome. String nucleosomes together, and you get chromatin. A beaded fiber. Chromatin coils into loops. Loops bundle into domains. Domains stack into chromosomes. But the packing isn't uniform. Some regions are tight, silenced, inaccessible. Other regions are loose, open, active. The material state of DNA determines what genes can be read. Experiments measuring chromosome bending stiffness found values around 10 to the negative 22 Newton meters squared. That's incredibly soft for a solid. For comparison, a steel wire of similar thickness would have a bending stiffness billions of times higher. DNA is floppy at the molecular scale, but when millions of nucleosomes pack together, the collective structure becomes much stiffer. Not steel, more like a gel, a weak elastic network that can deform and recover. Another team used a technique called loopsec to measure DNA bendability across an entire yeast chromosome. They tested over 270,000 50 base pair fragments. Each fragment's intrinsic bendability depends on sequence. AT, rich regions bend more easily. GC, rich regions are stiffer. The data revealed something unexpected. Regions just upstream of gene promoters often have unusually low bendability. Stiff DNA, that stiffness affects chromatin remodeling. Enzymes that slide nucleosomes around have trouble pushing into stiff linker DNA. Low bendability creates barriers. It helps define nucleosome-free regions, the zones where transcription machinery binds to start making RNA. Sequence encodes not just information, but also mechanical properties. A string of AT versus GC bases changes how easily the DNA bends, and that physical difference controls where nucleosomes sit, which genes are accessible, which are buried. Evolution didn't just write a code. It tuned the stiffness of the polymer to create mechanical signals the cell can read. July 2nd, 2025, UC Irvine. A paper in Nature describes a DNA element no one predicted. They called it range extender, REX. The discovery started with a question about enhancers. Enhancers are stretches of DNA that activate genes. They bind transcription factors, boost expression. Most enhancers work at short range, 10 to 50,000 base pairs from their target gene. Some work at medium range, 50 to 400,000 base pairs. A few work at extreme distances, over 400,000 base pairs, sometimes over 1 million. The question was how? What lets an enhancer reach across 1 million base pairs of coiled chromosome to find its gene? Evgeny Kavan and his team at UC Irvine focused on the sonic hedgehog gene, SHH, critical for limb development in mice. SHH has a long-range enhancer called ZRS, located 840,000 base pairs away. That distance is enormous in genomic terms. The Kavan lab took short-range and medium-range enhancers from other genes and moved them to the ZRS location, swapped them in using precision genome editing in live mice. The transplanted enhancers failed. They couldn't activate SHH from that distance. Even though those same enhancers worked perfectly at their native locations, distance killed them. Then the team looked at the DNA around the ZRS enhancer. They found a conserved element nearby. A short sequence with no enhancer activity on its own. It didn't boost gene expression when tested alone, but when they added this element to the short-range enhancers, something dramatic happened. Range exploded. An enhancer that normally worked at 71,000 base pairs suddenly activated genes at 840,000 base pairs, over 10 times farther, an order of magnitude increase. They named the element a range extender, REX. At the molecular level, REX contains short repeating DNA sequences. These sequences serve as docking sites for proteins that help form DNA loops. The cell uses protein complexes like cohesin to bring distant regions of chromosomes into contact. REX appears to recruit or stabilize these loops, making long-range communication possible. The team found that REX-like sequences are enriched around other long-range enhancers, genome-wide, including the ZRS itself. When they mutated specific motifs within the ZRS, short stretches with the pattern C or T followed by AATA, the ZRS lost its long-range activity. It could still activate genes at short distance. The cell type specificity remained intact. But the ability to work at 800,000 base pairs vanished. Mice with those mutations showed severe limb defects, missing forearm and hand bones, evidence that long-range gene activation is essential for normal development. The implications ripple outward. If range extenders are common, then many non-coding mutations linked to disease might disrupt long-range enhancer function without affecting short-range activity. A variant far from a gene might still cause problems by breaking a REX element, stopping an enhancer from reaching its target across hundreds of thousands of base pairs. That creates a new layer of genetic architecture to map, to understand, to screen for in medical genetics. It also means enhancers are modular. They have separable parts. One set of sequences determines which cell types respond. Another set determines how far the enhancer can act. Evolution can mix and match these modules. Add a REX to an existing enhancer, and suddenly it gains long-range capability. Remove a REX, and the enhancer becomes short-range only. This modularity accelerates evolution, provides flexibility. Lets genomes experiment with new regulatory strategies without breaking existing gene control. Now, consider what this means when you put it all together. DNA is a double helix, mostly, but it's also sometimes a four-stranded knot, or a left-handed spiral, or a cruciform junction. 8% of it came from ancient viruses that now help build your placenta and guide your embryonic development. It behaves like a solid when you pull it fast and a fluid when you pull it slow. Its sequence encodes both genetic information and mechanical stiffness. Hidden elements scattered through the genome let enhancers talk to genes across nearly 1 million base pairs. And huge regions once dismissed as junk turn out to be regulatory machinery, controlling when and where your genes turn on. The molecule isn't a simple instruction manual. It's a dynamic material, a library of evolutionary experiments, a battlefield where viruses and hosts fought for millions of years and then made peace. A scaffold that bends and loops and remembers its shape. The strangest part is how much remains unknown. Scientists finished sequencing the human genome in 2001, but that sequence had gaps, big ones. Centromeres and telomeres were missing. Repetitive regions were collapsed into averages. It wasn't until 2022 that the Telomere to Telomere Consortium published the first truly complete human genome. Every base pair, no gaps. Those newly sequenced regions are packed with non-B DNA structures, with repetitive elements, with HERVs and other transposable sequences, and each discovery in those regions reveals functions no one predicted. Viral DNA regulating gene expression, stiff sequences defining chromatin boundaries, G quadruplexes marking replication origins. The complete sequence opened a door to a genome we barely understand. Even though we've been studying DNA for 70 years, if you value documented genetics and molecular biology research like this, subscribe. The best way to support our work. Also comment from which city you're hearing this from. This script is researched and factual. Using sources from multiple peer-reviewed journals, including Nature Nucleic Acids, Research and Science Advances, published in 2024 and 2025, documenting direct experimental work on DNA structure function and evolutionary history conducted at UC Irvine Kyoto University, Princeton University Penn State, and Lawrence Berkeley National Laboratory. Real molecular biology from labs actively sequencing genomes and engineering chromosomes in living cells and organisms. Now, back to the information. Consider the timeline. Retroviruses started integrating into primate genomes at least 100 million years ago. Some HERV families are shared across all primates. Others entered later. The HERV-K-HML2 family is the youngest. It was still actively copying itself less than 5 million years ago. Some insertions are unique to humans, not found in chimps or gorillas. That means viral activity continued even after the human lineage split. These weren't ancient one-time events. They were ongoing, repeated, a continuous process shaping the genome. Every few generations, a new viral insertion. Most did nothing or caused harm and were eliminated by selection. But occasionally, one landed in the right place, near a gene that needed regulation, near an enhancer that needed range, and the viral sequence got repurposed, domesticated, made useful. The genome is full of these domesticated viral elements. Researchers estimate that roughly 45% of the human genome consists of transposable elements, sequences that can, or once could, copy and paste themselves, line elements, sign elements, DNA transposons, and endogenous retroviruses. Together, they make up nearly half your chromosomes. Only about 1.5% of the genome encodes proteins, genes in the traditional sense. The rest is regulatory sequences, structural elements, evolutionary debris, and repurposed viral machinery. What about the mechanical properties? That viscoelastic behavior measured at Princeton. Why does it matter? Because chromosomes need to reorganize during cell division, during development, during differentiation. An embryonic stem cell has its chromosomes organized one way, open, accessible, ready to activate any gene. A fully differentiated neuron has its chromosomes organized completely differently. Most genes locked down, silenced. Only neuron-specific genes accessible. That reorganization requires movement. DNA loops need to form and dissolve. Enhancers need to find new promoters. Chromatin needs to condense and decondense. If chromosomes were rigid, this would be impossible. If they were too fluid, it would be uncontrolled. The viscoelastic balance provides both stability and flexibility. Cells exploit this. They use it to fine-tune how fast regulatory changes happen. A quick signal can't immediately reorganize a chromosome. The elastic resistance prevents that. But sustained signaling over minutes or hours allows the chromosome to flow into a new configuration, which acts as a form of mechanical memory. The chromosome holds its shape until something pushes hard enough or long enough to change it. Researchers at Princeton demonstrated this using their light-controlled system. They used lasers to create condensate droplets at specific DNA locations. The droplets pulled DNA strands together, forced them into close contact. Then the researchers turned off the laser. The droplets dissolved, and they watched what happened. In some cases, the DNA immediately sprang back to its original position, elastic recoil. In other cases, it stayed in the new configuration. The contact was stabilized by proteins, by chromatin structure, by epigenetic marks. The mechanical manipulation created a new regulatory state that persisted even after the external force was removed. This suggests cells use mechanical forces to organize genomes. Motor proteins pull chromatin. Condensates form at specific sites and create compartments. Transcription machinery generates torsional stress that bends and twists DNA. All of these forces shape chromosome architecture. And that architecture controls which genes are on or off. Information and mechanics are intertwined. The discovery of non-B DNA structures adds another layer. G quadruplexes form in guanine-rich sequences. These structures are thermodynamically stable under physiological conditions. Potassium ions from the cellular environment stabilize the stacked G tetrads. Once formed a G quadruplex is hard to unfold. Helicases exist specifically to unwind them. Cells need machinery to remove G quadruplexes from DNA replication forks. Otherwise, replication stalls. But cells also use G quadruplexes as regulatory signals. Transcription factors recognize them, bind them, use them as markers for where to act. A study on SARS-CoV-2 found G quadruplexes in the viral RNA genome regulate translation. Small molecules that bind these structures inhibit viral replication. Similar principles apply to human G quadruplexes. Drugs targeting G quadruplexes are being explored as cancer treatments. Because many cancer-related genes, including MYC, have G quadruplex motifs in their promoters. Stabilizing the structure represses the gene. That slows tumor growth. At least in some contexts, zDNA is equally complex. It forms transiently during transcription. When RNA polymerase moves along DNA, it creates torsional stress. The DNA ahead of the polymerase gets overwound. The DNA behind gets underwound. Underwound regions with alternating purine pyrimidine sequences flip into zDNA. That marks the region as transcriptionally active. Proteins that bind zDNA recognize those marks and recruit regulatory machinery. ADAR1 edits RNA at those sites, converting adenosines to inosines, changing the RNA sequence, which changes the protein sequence or affects RNA stability. zDNA is a signal that transcription happened here. It links the mechanical act of making RNA to downstream regulatory decisions. Evolution found a use for an unusual DNA structure that arises naturally from the physics of transcription. Cruciforms and triplex DNA add more options. Cruciforms form at inverted repeat sequences, palindromes. The DNA folds back on itself, creating a four-way junction, shaped like a cross. These junctions can block transcription. They can serve as recombination sites. They affect chromosome stability. Triplex DNA forms when a third strand binds in the major groove of a double helix, usually at polypurine polypyrimidine sequences. The third strand uses Hoogs-Steen-based pairing, a different hydrogen bonding pattern from Watson-Crick pairs. Triplex structures have been found in human cells. They're associated with gene regulation, with chromosomal breaks, with sites where DNA repair machinery acts. Each non-B structure provides a distinct signal, a different shape the cell can recognize and respond to, and they're all sequence encoded. Specific base patterns favor specific structures, which means the genome contains instructions for forming these shapes in particular locations, for particular purposes. The MER 11 viral sequences studied by the Kyoto team exploit this. Those sequences don't just contain binding sites for transcription factors. They contain sequence patterns that affect DNA shape, bendability, accessibility. The viral DNA evolved to integrate into genomes, to persist, to spread. That required evading silencing mechanisms, staying accessible, being available for transcription factors to bind. The sequence features that helped the virus survive in the genome also make those sequences useful as regulatory elements. High GC content, nucleosome excluding motifs, flexible bending properties. The host genome didn't design these sequences. The virus did. But evolution in the host took advantage of the properties the virus already built, repurposed them, used them to regulate genes the virus had nothing to do with. It's not intelligent design, it's opportunistic tinkering, evolution grabbing whatever works and pressing it into service. The range extender discovery at UC Irvine reveals the same principle. REX elements aren't novel human inventions. They're conserved sequences found across mammalian genomes that suggest they arose early, possibly in a common ancestor. And the motifs they contain, those A-ATA homeodomain sites, are known protein binding sequences. Homeodomain transcription factors regulate development, pattern formation, body axis specification. The same proteins that tell cells where they are in the embryo also help enhancers reach across long genomic distances. That connection isn't accidental. Development requires coordinated gene expression. Genes in different parts of a chromosome need to respond to the same signals at the same time. Long-range enhancers provide that coordination. REX elements make it possible. Without them, development fails. The UC Irvine team demonstrated this directly. Mutate the REX motifs in the ZRS enhancer, and mouse embryos develop severe limb defects. No forearm, no hand. Because one gene, Sonic Hedgehog, didn't activate at the right time in the right place. Because the enhancer couldn't reach it. One small DNA element, a few dozen base pairs with a specific sequence. And it's the difference between normal development and catastrophic failure. That's the real strangeness of DNA. It's not one thing. It's information storage and mechanical scaffold and regulatory network and evolutionary record all at once. The sequence matters. The structure matters. The dynamics matter. Change one base pair and you might do nothing. Or you might break a REX element and lose long-range gene activation. Or you might create a G quadruplex where there wasn't one before and stall DNA replication. Or you might activate a dormant HERV and turn on a viral enhancer that changes when a gene is expressed. The genome is a high-dimensional object. Sequence, shape, dynamics, context, all interacting, all contributing to function. And we're only beginning to map the full space. The completed telomere-to-telomere genome revealed how much we'd been missing. Centromeres are massively repetitive, thousands of copies of satellite sequences tangled together. The old sequencing methods couldn't handle that. They collapsed repeats, guessed at lengths, left gaps. New long-read sequencing technologies changed that. Pacific Biosciences, HiFi reads, Oxford Nanopore ultra-long reads. Suddenly repeats became readable, assemblies became complete, and inside those repetitive regions, scientists found structure, organization, function. The Penn State team, mapping non-B DNA, used those complete genomes. Without them, the analysis would be impossible. You can't identify non-B motifs in missing sequence. The completed genomes showed that centromeres and telomeres are packed with G quadruplexes, with Z DNA motifs, with sequences predicted to form complex secondary structures. These aren't accidents, they're features. The high density of non-B forming sequences in centromeres likely contributes to centromere function, to how chromosomes attach to spindle fibers during division, to how sister chromatids are pulled apart, to how the genome transmits itself accurately from cell to cell. Telomeres tell a similar story. Human telomeres consist of thousands of copies of the sequence TTAG repeated over and over. That's a G-rich sequence, prime territory for G quadruplexes. Studies using laser tweezers to mechanically unfold telomeric DNA found G quadruplexes form readily. About 6 quadruplexes per full-length telomere overhang. Most don't interact with each other. They sit like beads on a string. But about 5% form higher-order interactions, stacking, clustering, creating more complex structures. The function remains debated, but evidence suggests G quadruplexes protect telomere ends, prevent degradation, regulate access by telomerase, the enzyme that adds new repeats. Mutations in proteins that resolve G quadruplexes lead to telomere dysfunction, to accelerated aging, to genome instability. The strange structures aren't optional. They're necessary. And throughout all of this, the viral DNA sits quietly, 8% of the genome, 60,000 insertions. Most are inactive, broken by mutations, unable to make virus particles. But the DNA remains. And cells read it, transcribe it into RNA, sometimes translate small fragments into proteins. The Kyoto study found MER11 sequences actively making RNA in stem cells, in early neural cells. That RNA doesn't become a virus. It doesn't do anything viral, but its presence affects which other genes are on. The viral enhancers boost expression of nearby genes, genes involved in development, in differentiation, in cell fate decisions. That means ancient viruses are still influencing how your cells behave, not by infecting them, by controlling them. The virus became a regulatory tool, a switch your genome flips to change cellular states. That's not junk. That's sophisticated control. The Human Genome Project took 13 years, launched in 1990, finished in 2003, cost $3 billion, involved labs across the world, and it delivered a draft. 95% complete. The remaining 5% took another 20 years, finished in 2022. Those gaps held secrets, functions no one anticipated. Now that the sequence is truly complete, the real work begins. Understanding what all those base pairs do, how they work together, how structure and sequence and dynamics create the regulatory logic that builds and maintains a human body. 46 chromosomes, 3 billion base pairs, 8% virus, 45% transposable element, 1.5% protein coding gene. The rest is regulatory, structural, functional in ways we're still discovering. Consider one more angle, DNA repair. Cells constantly fix damage. UV light from the sun causes thymine dimers. Reactive oxygen species break DNA strands. Replication errors introduce mismatches. Cells have elaborate machinery to detect and repair all of this. But non-B DNA structures complicate repair. G quadruplexes can block repair enzymes. So can Z DNA, so can cruciforms. The unusual structures create barriers, make certain types of damage harder to fix, which means those regions accumulate mutations faster, become hotspots for change. Evolution exploits this. Regions with high non-B DNA density evolve faster, adapt faster, provide raw material for selection to work with. But it also creates vulnerabilities. Cancer cells often have mutations in non-B regions, where repair failed, where the strange structures prevented the machinery from reaching the damage in time. The same features that drive innovation also drive disease. DNA's strangeness cuts both ways. The most recent discovery, range extenders, highlights how much remains unknown about basic genome organization. Scientists have studied enhancers for decades. They knew long-range enhancers existed. They knew enhancers, and promoters needed to physically contact each other. They knew about DNA looping, about cohesin, about chromatin architecture. But they didn't know about REX. They didn't know a specific sequence element was required for extreme distance activity. It took sophisticated experiments in genetically engineered mice to reveal it, replace an enhancer, test function, fail. Add or X, test again, succeed. That kind of experiment is only possible with modern genome editing tools. CRISPR, homologous recombination, precise insertion and deletion. 20 years ago, you couldn't do it. 10 years ago, it was cutting edge. Now it's standard, and it's uncovering principles that have been there all along, encoded in the sequence, waiting for someone to ask the right question. Princeton's mechanical measurements add another dimension. DNA is a material. That sounds obvious, but the implications are profound. You can measure its elastic modulus, its bending persistence length, its viscosity. Those physical properties affect biological function. A stiffer DNA region resists nucleosome formation. A more bendable region favors it. Sequence determines mechanics. Mechanics determine structure. Structure determines what proteins can bind, what genes can be accessed, what regulatory states are possible. The cell doesn't just read DNA as code. It feels DNA as material, measures its resistance, responds to its shape. That adds a whole layer of regulation beyond transcription factors and binding sites. Physical properties matter, and they're tunable. Evolution can adjust sequence to adjust mechanics, to create barriers or signals or flexible joints, all without changing the genetic code, without altering protein sequence, just by changing how the DNA itself behaves. The convergence of these discoveries paints a picture. DNA is not a static blueprint. It's an active, dynamic, responsive molecule. It contains instructions, but it also contains structure and mechanics and evolutionary history. 8% virus means your genome carries a record of ancient plagues. Non-B DNA means the molecule can take multiple shapes, each with different functions. Viscoelasticity means chromosomes remember and respond to forces. Range extenders mean long-distance communication is engineered into the sequence. Domesticated viral enhancers mean ancient infections now control your development. Every piece adds complexity. Every piece reveals that DNA is stranger than the double helix. Stranger than anyone imagined when Watson and Crick built their model in 1953. They got the core structure right, but the full story is so much weirder. Here's the kicker. We're still finding new things. The completed genome is 5 years old. REX was discovered this year. The Kyoto viral enhancer work published 6 months ago. Princeton's mechanical measurements are 18 months old. The field is moving fast. New sequencing technologies reveal new regions. New genome editing tools enable new experiments. New imaging methods show chromosomes moving in real time. Each advance uncovers functions that were always there but invisible. The genome you inherited from your parents is the same genome your ancestors had, the same DNA, but our understanding of what it does is accelerating. Every year brings discoveries that rewrite the textbooks, that reveal the molecule is stranger and more sophisticated than we thought. The practical implications are enormous. If viral DNA regulates early development, then mutations in those regions could cause developmental disorders. If range extenders control long-range enhancers, then disruptions could explain genetic diseases with no obvious cause. If non-B DNA structures mark replication origins or regulatory boundaries, then drugs targeting those structures could treat cancer. The strangeness of DNA isn't academic, it's medically relevant. It's the frontier where molecular biology meets human health. Understanding these features means understanding disease, designing better therapies, predicting genetic risk more accurately. It also means confronting how much we don't know. How many functions remain hidden? How many layers of regulation we haven't even suspected yet? The human genome contains approximately 20,000 protein-coding genes. That's fewer than a grape, fewer than a roundworm. The complexity of humans doesn't come from gene number. It comes from regulation, from how those genes are controlled, when they turn on, where they're expressed, how strongly. DNA's strangeness is largely about regulation, viral enhancers, range extenders, non-B structures, viscoelastic dynamics. These are all regulatory mechanisms, ways to control gene expression without changing the genes themselves. Evolution has been working on this control system for billions of years. Tinkering, adding layers, repurposing old parts, keeping what works, discarding what doesn't. The result is a genome that's astonishingly complex, not in its coding capacity, in its regulatory architecture. And that architecture depends on the molecule's strange properties, on its ability to fold into unusual shapes, to resist or yield under force, to carry both instructions and history. The viral DNA adds yet another dimension. Those sequences didn't evolve to help humans. They evolved to replicate themselves, to spread through genomes, to persist. But in doing so, they acquired features useful to the host: regulatory motifs, enhancer activity, structural elements. And over millions of years, the host genome integrated those features, made them essential. Now we can't develop properly without viral DNA, can't build a placenta, can't organize chromatin correctly. The virus became part of us. Not by accident, by evolutionary appropriation. The host took viral machinery and repurposed it for non-viral functions. That's not cooperation, it's exploitation. The host uses dead virus parts as regulatory tools. And it works because natural selection doesn't care about origin. It cares about function. If a viral sequence helps the organism survive and reproduce, that sequence gets kept passed down, integrated into the regulatory network. The fact that it came from a pathogen is irrelevant. Now think about the timeline again. Humans and chimpanzees diverged about 6 million years ago. HER-VK, HML2 was still active then. Some insertions are human-specific. That means viral activity continued shaping the genome even recently, even within the past few million years. Not ancient history, recent evolutionary time, and those recent insertions are more likely to be functional. Less likely to have accumulated disabling mutations. The Kyoto team found MER11G4, the youngest subfamily, has the strongest enhancer activity. Younger equals more active. That suggests a continuous process. New viral insertions provide raw material. Selection tests them. Most fail. A few succeed. Those few get incorporated into regulatory networks, start controlling genes, become essential, and eventually become invisible. Part of the normal genome, no longer recognizable as foreign. That's what 8% viral DNA represents. Millions of years of this process. Infection, integration, selection, domestication. Repeat. Layer after layer of viral sequences now woven into the regulatory fabric of the human genome. The Penn State non-B DNA study revealed another pattern. These unusual structures are enriched in newly sequenced regions, centromeres, telomeres. Paracentromeric repeats, the regions that were gaps in earlier genome drafts. That means the strangest parts of the genome were invisible until recently, and they're functional. Centromeres are essential for chromosome segregation. Telomeres protect chromosome ends. The non-B structures in those regions likely contribute to function, perhaps by recruiting specific proteins, perhaps by providing structural rigidity or flexibility, perhaps by marking boundaries. The full picture isn't clear yet, but the correlation is strong. High non-B density and functionally critical repetitive regions. That's not coincidence. That's biology using strange DNA structures to accomplish tasks that normal B-form DNA can't. The range extender story ties this together. Rex elements contain conserved motifs. Theta homeodomain sites. Those motifs bind transcription factors involved in developmental patterning. The same factors that tell cells their position in the embryo also help enhancers reach across long distances. That links spatial information in the developing organism to spatial organization in the genome. The enhancer needs to span 1 million base pairs. The Rex provides that capability by recruiting developmental transcription factors, which are only present in the right cells at the right times, which means long-range enhancer activity is restricted to specific developmental contexts. The genome uses developmental signals to control its own 3-dimensional architecture. Position in the embryo determines which chromatin loops form, which determines which genes can be activated, which determines cell fate. It's an integrated system. Body plan and genome organization linked through shared transcription factors and sequence motifs. DNA's strangeness enables that integration. Without Rex, you can't have long-range developmental enhancers. Without long-range enhancers, you can't coordinate gene expression across large chromosomal domains. Without that coordination, development fails. The molecule's unusual features are essential. One final twist. The Princeton mechanical work showed that chromosomes flow under sustained force. That flow is driven by motor proteins and regulated by ATP hydrolysis, active process, energy consuming, which means cells expend resources to organize their genomes. It's not passive, it's not just thermodynamics. The cell actively shapes its DNA, pulls loops, forms compartments, maintains structures. And those structures determine gene expression. So gene expression isn't just about transcription factors finding binding sites. It's about whether those binding sites are accessible, whether the enhancer can reach the promoter, whether the chromatin is open or closed. DNA's mechanical properties determine accessibility. The cell controls those properties using energy and motors and architectural proteins. That makes gene regulation a mechanical problem as much as a biochemical one. You can have all the right transcription factors present. If the DNA is in the wrong physical state, the gene stays off. The molecule's material nature matters. 7 decades after Watson and Crick published their double helix model, we're still discovering fundamental features of DNA. 8% viral, multiple structural forms, viscoelastic dynamics, long-range regulatory elements. All of this was there in 1953, encoded in the sequence, but invisible without the tools to see it, without complete genomes, without single-molecule mechanics, without precision genome editing in living organisms. Each technological advance reveals another layer, and every layer shows the molecule is stranger than we thought, more sophisticated, more integrated into cellular function, not just a passive code to be read. An active participant in everything the cell does. Information and structure and mechanics and history all intertwined. The future will bring more discoveries. Better imaging will show chromosomes reorganizing in real time. Better sequencing will reveal variation across individuals. How do range extenders differ between people? Do viral enhancers vary? Are non-B structures polymorphic? All of this likely affects disease risk, affects drug response, affects how individuals develop and age. Personalized medicine depends on understanding these features, not just gene sequences, regulatory architecture, structural variants, mechanical properties, the full complexity of the genome. And that complexity is strange, wonderfully, necessarily, functionally strange. DNA evolved over billions of years. It carries the history of that evolution in its sequence and structure. Viral invasions, transposon activity, mutations, selection, all written into the molecule we inherit. And that molecule uses its strangeness to build and maintain life, to regulate, to remember, to adapt. It's more than a blueprint. It's a dynamic material system with emergent properties that enable everything biology does. That's the truth. No textbook quite captures. DNA is strange, stranger than you imagine, stranger than the double helix. And understanding that strangeness is understanding life.